we're back on. Okay, so based on the fundamentals of molecular and differences in prokaryotes, the scientists have devised this group, which is the domains, okay? And the whole point, just to summarize all, it was no different than anything else. It's when we get these really cool bells and whistles, and we're still even divided. That's where the term supers and subs came up too, because they really started seeing that this big group of these animals that all look alike, wow, there's more diversity in that than we believe. Therefore, we don't have to redo the system. We have to add extra steps, if that makes, I mean, that's the easy way of summarizing why they do what they do now with the systematic approach, okay? Okay, so, which real quick review on that. I mean, I don't know why I threw that in there, but I'd hope to shout by now you understand we're all here. This is what I was just talking about, prokaryotes. When I'm talking about all anything that's a bacteria, anything that is single-celled with no membrane-bound nucleus inside, okay? It's just basically a nice little membrane, a little bit of cytoplasm or cytosol, whichever you want to call it, a couple little important organelles, and some random molecular material, DNA, okay? Although like us, you know, we have the fancy, you know, nucleus, which is the brain, houses all that. You know, if you go back to your earliest days of biology where you did your little cell city thing, maybe you made a cake or you made a cookie or, you know, something of that, some type of model. We're all in that. A lot more membrane bound, and membrane bound gives us a lot more attention to detail. So that's just real quick. You know, when you hear prokaryote, I call them just pros and U's, or P and E sometimes, to just abbreviate them, okay? Okay, so and they're not that pretty and colorful. I added color to the pictures. I mean, they're like transparent black and white. These are just various bacteria. I think that's salmonella, actually. And that's just, I think that's my salmonella one I got, a sample one. Okay, and cool little prokaryotes, you know, obviously that's a plant, that's an animal, that's a protista, and there's a toadstool mushroom, there's a fungi, all in those. So that's why they were showing that, just to show you some pictures, okay? All right, so now we call it a six-kingdom system, okay? Six-kingdom system. Most biologists use a six-kingdom system of classification. Two prokaryotic systems, or two prokaryotes and four eukaryotes. Sometimes, very rare, you may hear an old school professor, when you get to your next level of education, if you're in that, that's been around a while, he may refer to, or she may refer to, a five-kingdom system because they're accustomed to that. Or they'll say, yeah, no, five, and the old school six is still the same, blah, 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 okay? Or you may come across a textbook. Okay, probably not now with the editions, but if you had an older edition one, they might refer to it. But as you know, we got the two prokaryotic ones, those are the domains, archaean and new bacteria. And then you just showed you the picture of the four eukaryotic kingdoms, which are the plant, animal, protista, and fungi, okay? So we call it now six-kingdom system. I'm sure that's 99% of the time that's what you guys have been referred to as, because it has been set up in that system in the text and stuff for quite some time, okay? All right, so there's a picture, meaning I showed you kind of under electron microscope. This shows you where you find that. Pretty barren, not some place you want to hang your hat, you know. So when they mean really hostile type environments where there's a lot of maybe pyroclastic activity, geological phenoms, maybe plate tectonics, volcanic activity, deserts, you know, old school stuff. You know, that's why it makes sense where you find the, you know, ancient or archaean bacteria. Okay, some of the characteristics. Then here's our 5 little bullets that these are the basics. If you were a scientist and you had all the technology, this is how you would start, by just putting them in these broad groups. Okay. So one of the first things that have to be obviously is unicellular, prokaryotes, but this is the big one. Their cell walls, okay, do not have this structure called peptoglycan. And if you are glycogen or glyco refers to like a sugar-based type stuff, if you were a person who was a microbiologist and all those type of things, you would know what to look for with this, okay? But that's the main thing that basically separates those two groups of bacteria, is the absence of or presence of this peptoglycan, all right? They differ a little bit biochemically from eukaryotes, and then this is kind of where you find them. When they talk about hostile environments, just a lot of really, you would probably find a lot of ancient bacteria like the cool pools like at Yellowstone you see, you know, those sulfur looking things, and the various different real ornate, colorful looking ones because of the minerals and bacteria that are growing, halophiles and things of that nature, which are bacteria that grow in extreme temperatures and stuff like that. That's where you probably see that kind of stuff. Deep water fissures where have in the ocean where they're bubbling up lots of gases and stuff. If you were a deep sea, so areas like that. Chances of us coming across these archaean bacteria, you'd have to be out on that field of study. Maybe in an old lava field in Hawaii or something of that nature. So deep dark caves, so that type of area. Okay. Here's just your good old-fashioned bacteria, a simple picture there under electron microscope, threw in some coloration there. So, but basically with your normal bacteria, oops, hit the wrong screen, is characteristics are the same thing, unicellular, prokaryote, but here's that big thing. They generally have peptoglycan in their cell wall, okay? All right. Maybe, and they're also, since they're more common and they're the new bacteria, as we know, bacteria are very, very important decomposition or decomposers, important for breaking down organic material. We've got to have that. A lot of them are photosynthetic because, you get in them dank, dark caves and things of that nature. And they can't do, they do photosynthetic, but then areas where it is really dark, they use chemosynthetic or chemosynthesis. They can take chemicals and convert them because the absence of light, okay? So it's an adjustment. So a lot of those like bioluminary type deep water fish and algae and things like that would be using chemosynthesis type stuff, that are down deep below, out of the reaches of lights, dark caves, things of that nature. And even many of them are parasitic or parasites, okay? All right, and that Giardia is one of them. Amoebocytes are another one. Okay, but a lot of them also can be parasitic and harmful. Right? Parasite. Remember the definition. If you don't know what a parasite is, it's a symbiotic relationship in nature, meaning parasitic feeds off another host, needs another host, feeds off another host. Okay, but doesn't benefit. Okay, so it's not a good thing. Okay. Okay, so we got our protistas, the ones that really get a bad rap. They're usually thought of as fresh water, most of them, water-based little structures. I guarantee if you've been to a pond and you pond water, or maybe back in your days at grade school where you looked under a light microscope, you might have seen one of those little euglenas or protozoas or things of that nature. They look like little elongated with little cilia around them. Those were in those categories. Okay, so they've been nicknamed the catch-all kingdom. And basically what it means is because they never really had a place, okay? They were eukaryotic, so they definitely weren't a bacteria. But a lot of them were unicellular, so that's what really, they couldn't be a plant or an animal. Or in a lot of cases, they also could not be a fungus because most funguses are multi-cell. There are a few, the exceptions, okay? So that was the key thing, is they're eukaryotic, but unicellular. Some were simple, but anyways, without going into a lot of detail, the scientists, it was always clear that, this is a plant, this is an animal, this is a bacteria, this is a fungus, and then you're like, let's put this one over here, we'll come back to it later. Let's put this one over here and come back to it later. And if my numbers are right, I want to say there's like 4,000 to 6,000 last I looked. But they started collecting this large group of, okay, we got to put them somewhere. So that's where the term protista, which means catch-all, the things that all kind of fall, the tweeners. So that's where they call it the catch-all kingdom, those things that kind of have not the perfect place like all the others do. So that's why they call it that, okay? Funguses. That's actually the top of a cut-down tree that you might have seen like that, where they grow on the sides of trees and stuff, plant fungus. This is actually fungus in athlete's foot, I believe. I took that little picture. And that's actually the nape of somebody's arm. Have you ever had ringworm? That's ringworm. Ringworm is not a worm, it's a fungus, okay? It's just from getting real hot and sweaty, your pores get real open, dirt and bacteria can get in there and causes this. Little cortisone cream, clears it up type stuff. Real common, and wrestlers that get it all the time. Sometimes, I remember one year I had a catcher years ago that he had the worst case of ringworm, to the point to where we literally laid his personal catching gear down and sprayed it down with bleach. That's how he was getting. He was a catcher, so his gear was all stinky and stuff. So he was always getting it, and then he'd get it cleared up, and it'd come back, clear it up, come back. So, but yeah, it doesn't like make you feel bad or nothing. It's just annoying. So, all right. So a fungus is, notice, eukaryote, the same, usually multicellular. All right. Here's the cool thing is they're heterotrophic, because a lot of times people are, well, aren't funguses just plants? No, because they're not, because they eat things. They have to eat something to get energy. They don't make their own energy. So that's a big factor, how they absorb their nutrients. All right. And they also have this structure called chitin, which is kind of a crunchy type. If you ever take like a fresh mushroom and go and pop it, okay, and it has those little striated little strings. If you ever seen that and that come off, these are these Hypea type stuff, which are just kind of like little fibers that give them a little, they're flexible, but gives them some stability to it. Okay, so these are just some of the things with the fungi, but this is one of the big reasons why they're not lumped into plants, because they have to eat things. That's, you know, in order to feed on it. Pretty plants, various different ones, yada yada yada. Okay, they're eukaryotic, multicellular. Their cell walls don't have chitin; they have cellulose. Okay, and they also have structures called chloroplasts, because that makes them photosynthetic. And they're autotrophs, meaning they make their own food. So that's an easy one there. Okay, so there's not a lot of fancy details that put these structures in. What's the word I'm looking for? Fancy structures to put them in the groups. Okay, so a lot of times you think there's a lot of really elaborate details. There's really not. Okay, I mean, you got to start somewhere, and then from there you break them down from there. Okay, okay, and animals. Okay, and I thought I'd put that up as a joke. There's no worm that looks like that. Hopefully not, because I'll never touch a worm again if there's one crawling around up there that looks like that. I don't remember why I put that one on there, but I just did for the fun of it. Okay. So, and of course, we're in the animal kingdom. We're eukaryotic. I mean, so technically, we're multicellular. We have no cell walls. Heterotrophic, ingest food. Biggest thing is most have some type of simple to complex nervous system. And there are other things that fall in those parameters, but for this purposes, we're not going to extensively in that. Just showing you how easy it really is to put these things in these respectable groups. Okay, the animal kingdom is very, very broad. Okay, it's some of you might be hard to fathom that you're like, yes, we're in the animal kingdom. So is a starfish, huh? We don't look anything like a starfish. Well, there's so many little factors, and they're so broad that we have in common. Like, for example, starfish goes all the way back to how their germ layers, like when their little tiny cells, develop, which puts them in the category in the animal kingdom, which goes, okay, if you're a person that studied invertebrates and that stuff, you would know this stuff. And when we get into that one invertebrate phylum later on, first of the year, whatnot, we'll cover that. It's called a term called deuterostome and protostomic. Okay, and things that are deuterostemic fall into the animal kingdom, so it's kind of interesting. So anyway, I don't expect you remember that, but it'd be real cool if you did. All right, so I always say this: phylogeny or phylogenetics or phylogenetic trees. You hear all those same type of terms. Are basically a term associated with evolutionary relationships of organisms. Okay, remember the term? How are they related over time? Remember the term evolutionary type stuff. So a lot of it, the systems in which how we classify things, all right, attempt to reconstruct a phylogenetic tree. Okay, we know that as, you know, we got our reptiles, we got our birds, we got this, we got that. This goes a little more specific. Okay, so they really take a group within a group and they break those down internally from there. Okay, classification of organisms is based on common ancestry. So they'll take a common ancestor at the base of the trunk, and then they will show all the various different avenues. Like you could take the family Ursidea, bears, okay, and go back to the earliest bear, whatever it is, of the trunk, and then you could go all the way off various things, all the way from your lesser pandas and koalas to your grizzlies, Kodiaks, you know, that's what I mean by that. You can take an individual group and then you show the species or the detail within that particular family of animals. Okay, think something like that. That's what it means by based on a common ancestor. All right. I think I even have it. But here's probably one of the most important slides in there that it really doesn't seem. This right here, people, is what has really changed the game. What do you mean? Molecular? Well, yeah. Because when Linnaeus started this, okay, in the binomial system, pretty much everything was structurally what you saw: horns, hair, color, even toed, gnawing animals, whatever it might be. It was visualization. Nothing wrong with that, but there were some things that were put in places there shouldn't have been. Physiological is how the things work. Like, for example, herbivores, okay, non-plant eaters, carnivores, meat eaters. That's a physiological. There was even behavioral. This wasn't mostly it was these two, but then you threw in the molecular. DNA came in the picture. That cleaned up in a lot of organisms being misclassified, or it didn't necessarily clear up more organisms that were being misclassified. What it helped clear up was looking at one organism thinking, wow, it's because we visually would look at it, we thought they looked like a certain organism. Turned out they weren't, because DNA is real simple. The more patterns in your DNA profiles, when you put them next to each other, it's like almost like Pictionary. The more sequences they share in common, the more closely they are related. They're on the phylogenetic tree. That's just common sense. Okay, so there were certain organisms when they compared them, they realized that were not more any closer together than we thought, but they looked like it, but they really weren't. Okay, so that was one of the biggest things. Homologous means similar. Presence in two or more species, structures derived from a common ancestor. Could be anything from paired fins, claws, appendages. I mean, it could be a lot of different things. Okay, but there's a similarity structure to that. Okay, but these right here: structural, physiological, behavioral, and molecular are the four reasons and how we evaluate, put things into the groups all the way down from domain now, all the way down to species. Okay. All right, so here's what I'm saying. The more closely related two organisms are, the more characteristics they will share, the more classification categories they will share. Okay, so this is really broad, meaning are they in the same order? Are they in the same family? Are they in the same class? That type of stuff. Okay, so every time you go down a level from domain down, you're getting think of the pyramid now as not upright, it's upside down. I'm going broad, domain, kingdom, phylum, class, order, family, genus, species, all the way down to one particular group. Okay, so that's what's going on now. All right. So, for example, here is humans compared to cats. Okay, I just picked two random ones since we already had humans in it. We're obviously from the same animal kingdom. We have the same phylum Chordata, which means spinal cord. We both have like a hollow nerve cord for spinal cord. We're both mammals, obviously. So then, even though we're carnivores, there's other details. We come from the order primate, but this was a broader one, carnivora, meat eaters, although we're omnivores, not just true meat eaters. Okay, so we have three of the basic, 7 basic ones, the true Linnaean system. I mean, we could call it 8 if we threw the domains, because we're both from the domain Eukarya. I mean, if you wanted to, you don't have to, but we're talking about kingdoms. But you can see, and as we get really good, when we get at the latter end of this unit and we get into our semester project, you guys are going to be experts on all the hands-on stuff that we do, the phylogenetics, cladiograms, and everything. Okay, this is just laying out the content, understanding of everything, how it works. Okay, so it's just an example I gave you. So similarities do not always mean a relationship. Okay, so similarities may be the result of what they call convergent evolution. Things come together. Okay, this occurs when two species develop similar characteristics because they live in similar environments. Can anybody think of something like that? Ocean. Ah. How about like animals that live up in the canopy, or animals that live in burrowing type ground? There's certain features that they share in the same type of ecosystem. That, yeah, they're clearly different, but they have some because they need that in that environment. Here's a great example of it. Probably the most common one when they talk about homologous structure. Here's a yellowfin tuna, shark, and flipper the dolphin. I mean, yes, clearly 3 different species. Okay, actually that's a marine mammal, that's a chondrichthyes, a cartilaginous fish, and that's a osteichthyes, that's a bony fish. So actually, they're 3 completely different classified organisms, but they look a lot alike. We call them, just everybody probably just want to call them fish. Okay, but they're not. That's a shark, that's a fish, that's a marine mammal. But you see they got fins, the streamlined dorsal fins, pectoral fins, caudal fins, okay, and so on. So that's what it means by convergent. Because you live in the same environment, they have similar structures that look similar to everything else, okay? Let's see real quickly. 17. Okay. So this is where I wanted to leave off. But here's an important thing to understand. We're going to get into these things called cladiograms. Probably the first time you guys will see them, since you got a quiz tomorrow. I'll probably make a little bit of the lecture for that. It won't be very long, whatever. We'll see where we're at on time when I start it. I don't know how long it'll take you guys to take your quiz or whatnot, but we're only here. If you had a block schedule, it'd be easier. But we're going to start talking about what they call ancestral and traits, or shared ancestral and derived characteristics. These are the things that help separate out and how we divide them up along ancient to more modern type structures. So basically, the last two little slide here, and then I'll shut it down here. Shared ancestral characteristics are features that were present in an ancient species that remain present in the group today. Real simple, okay? For example, a vertebral column, vertebra, spine, cord, the bones, okay, is an ancestral characteristic found in all vertebrates. So what that means is, if it's an ancestral characteristic, we can't use that characteristic to separate out A from B. That's what that means, because all of them have it. Okay, so they can change how they're categorized depending on what I'm comparing, and that will make sense. All right, we cannot distinguish, like I said, between members of a group using an ancestral characteristic because they all have it. Okay, all right, okay. So, and that will leave us off there at derived characteristics, which is the one that gives us all our diversity. All right, and I'll review this again next time, because these are main points that we will refer to a lot with some of the exercises we do. Derived characters are traits found in two or more taxons that first appeared in the most common recent era. What do you mean? All right, for example, 3 small bones in the ear are useful in identifying a branch. So if you were a paleontologist, a zoologist, somebody who studies animals, and you have an organism that you're unable to, you might be able to, with a CAT scan, or if it's a bone, or if it's a skeleton or whatever, identify, you would look for this key thing. And for example is, these 3 tiny bones, we have them. Anybody know the names of them? The common names? You ever heard of the anvil, hammer, and stirrup? It's in the middle here, the 3 tiny little bones that pick up vibrations, okay? But anyways, this would be considered a derived characteristic when we were identifying, separating reptiles from mammals. Because mammals have them, reptiles don't. So they could be called a derived trait. But in mammals, it would be an ancestral trait. Does that make sense? Because I can't separate mammals with it, but I can use that feature to tell if it's a reptile or mammal. So that's what I'll explain, how sometimes, depending on what I'm comparing, can sometimes depend if it's a derived or an ancestral characteristic. Okay? All right, so I'm going to stop there with that. Okay. That's a big point with that.
Thanks for being patient.